If you need an interesting story about boosting insulin production in type 1 diabetics, we have a company to highlight for you today. It's called Persista Biotechnology, and wait for it, these guys think they can do it without immunosuppression drugs. I have to hear more about it. What about you?
Welcome to Type 1 Diabetic Warriors. Our team is committed to bringing Type 1 diabetics the latest medical developments to manage this disease. We also cover current medical advancements seeking a cure for T1D. If you value this content, please feel free to show your support by clicking on our homepage and hitting the link to buy us a coffee. Your support helps our team to research topics for upcoming videos. We thank you for your support.
Persista Biotechnology has developed an implant to deliver extra oxygen that will restore insulin production. Encapsulated islet cell implants, a biotech startup, Persista Bio, with roots at Cornell, is developing an implant that restores insulin production without requiring immunosuppressant drugs. This technology works by delivering extra oxygen. What is this about? Here's a breakdown of what this technology from Persista Bio is all about. What problem it's trying to solve, how it works, and where it stands right now. A promising solution is cell therapy, which involves implanting new insulin-producing cells derived from stem cells or donor islets to mimic the function of a normal pancreas. But there are big challenges. Oxygen supply. Implanted, encapsulated cells often don't get enough oxygen, especially if the cell-dense device is large or poorly vascularized. Blood vessels don't grow into it fast enough. Without enough oxygen, the cells die or lose function. Many earlier encapsulation devices had either poor oxygen transfer, poor immune protection, or both. Even if immune protection was good, the cells in larger or denser implants would become hypoxic, low oxygen, lose function quickly. Persista Bio, a startup with roots at Cornell's licensing from Cornell's Giner Labs, has developed what they call the O2 line system, or sometimes oxygenated macroencapsulation, or continuously oxygenated macroencapsulation, to try to solve both immune protection and oxygen supply, encapsulation and immune protection. The insulin-secreting or stem cell-derived islets are placed inside a capsule, a cell pouch, or macroencapsulation device. There is a semi-permeable membrane, nanofibrous shell that lets nutrients, glucose, insulin pass through, but blocks immune cells and large proteins, so the body doesn't reject the therapeutic cells. Implanted oxygen supply. This is the novel bit. The device includes a miniaturized implantable electrochemical oxygen generator. This generator uses water and moisture in the surrounding tissue and electricity, electrolysis, to produce oxygen. This oxygen is delivered to the encapsulated cells so they don't starve of oxygen and can function well. Designed to maintain function over time. The capsule is designed to allow high cell density, lots of insulin-producing cells packed in, but still maintain oxygen and nutrient diffusion. It also uses materials co-membranes aimed at minimizing fibrosis, scar tissue, and immune reactions that can degrade function. Proof of concept, animal testing. In preclinical, animal studies, in diabetic rats, this system was able to reverse diabetes, normalize blood sugar, without immunosuppression for up to 90 days, 3 months. Scalability and next steps. They're planning studies in large animal models, like pigs, working toward human-compatible versions, manufacturing under GMP, etc. Most encapsulation systems rely entirely on passive diffusion of oxygen from surrounding tissues. But oxygen diffusion is limited. It drops off with distance. Implanted devices often don't have enough blood vessel growth immediately. Encapsulation membranes or fibrotic responses can further restrict diffusion. Safety. Any implantable device has a risk of infection or mechanical failure. Also, ensuring that the immune barrier doesn't leak or that cells don't provoke immune activation. In August 2025, this channel reported that Cornell plus Giner Labs published a paper in Nature Communications showing the effectiveness of the system in rats. Persista Bio is still in the preclinical stage, moving toward large animal, porcine models, and working on human-compatible prototypes. They have secured NIH SBIR grants to scale up, validate in large animals, and do GMP manufacturing. If all goes well, this kind of technology could be transformative. A functional implant that restores insulin production without needing immunosuppressive drugs. Reduced need for constant blood sugar monitoring, insulin injections, pumps. potentially fewer long-term complications of diabetes, which are largely driven by imperfect glucose control and fluctuations. Scalability. Using stem cell-derived islets could allow widespread treatment rather than depending on rare donors. Well, what do you think?
Well, if you can boost the production of insulin in type 1 diabetics, I think you're pretty much almost there as far as solving the problem. If you can do it by oxygenating the transplanted islet cells, thus keeping them alive, I think you've got a home run. They're in preclinical trials, so they've got a way to go, but I love the idea. I never thought that oxygen would play such a big role in keeping the transplanted cells healthy, but apparently it does, and we wish them all the best. We'll have another important video for you here on Type 1 Diabetic Warriors in just a few days. Have a good one.